Welcome back, everybody. Having discussed the DNA repair, now we will be going into mutations in DNA and transcription. Starting with mutations in DNA. Degree of change: silent is less than missense, than nonsense, than frame shift. Frame shift mutations. What does this mean? This means that the frame shift mutations leads to more extensive damage rather than a silent mutation. What are the silent mutation, missense, nonsense, frame shift? We will discuss later on in coming videos. Now we will go to single nucleotide substitutions are repaired by DNA polymerases and DNA ligase. Type of single nucleotide point or point mutations are transition and transversion. Transition include purines to purines, pyrimidines to pyrimidines. Transversion is change of purines to pyrimidines or either change of pyrimidines to purines. Now we will go into single nucleotide substitutions, then we will go to other mutations. First, coming to single nucleotide substitutions, there are silent mutations, missense mutations, nonsense mutations. Silent mutations codes for same synonymous amino acids often involve third position of codon, tRNA verbal. What does this mean? See, we have already discussed in Verbal's hypothesis that the third amino acid is not important, or which is known as degeneracy of a code, which means the first two amino acids, see in a codon there are three amino acids, let's say A, C. The first two amino acids are important. The third amino acids will not be much, we don't bother much about the third amino acids. All we bother about the first two amino acids. So if there is a mutation in the third amino acid, it we does not care. See, in a codon there are nucleotides which code for amino acids. So, in codon there are nucleotides. So, if the third nucleotide of a codon is mutated or not mutated, we will get the same amino acid over there. So, we is the least bothered. So, in silent mutations there will be least amount of damage. In missense mutations, what happens is there will be other amino acid will be produced. See, we'll read it first. Results in changed amino acid called conservative if new amino acid has similar chemical structure. Examples include sickle cell disease, substitution of glutamic acid with valine. What they are what they are going to tell is, see, if we have amino acid from a codon and there is another codon which gives rise to other amino acid. See, there is a change in amino acid. This is known as missense mutation. In silent mutation, there is no change in amino acid, but here there is a change in amino acid. Whereas in nonsense, there will be no production of amino acid. How does it happen? We'll see. Results in early stop codon. UGA, UAA, UAG usually generates non-functional protein. Stop the nonsense. See, these are known as stop codons. On transcription, if this is the codon that your transcription encounters, then what will happen is your transcription completely stops. So these are known as stop codons. How to remember this? You are gone away, you are gone away, you are away, you are gone. So this is the mnemonic which will be dealt in the further pages. See, there are this. So in these three types of mutation, there is change in only single nucleotide. That change in single nucleotide is bringing about the mutation. But in other mutations, in frame shift mutations, what will happen is, let's say A C G C A C U. So this, let's say this is an chain, a chain of nucleotides. Now let's say this has been removed. Now what will be the codons? Let's say the codon. These three will be codons. These three will be one codon. And I will add another nucleotide. So this will be another codon. Let's say we have lost our guanine. What will happen? AAC, CAC, UAC. The code has shifted. First three nucleotides are okay. The sequence is okay, but the next three has become CAC. Here it is GCA. So there is a complete change in the reading of an nucleotide chain. So this results in either deletion, either the change in the sequence of amino acids, or there might be a stoppage in in the transcription. So this all might happen. First, we will see deletion or insertion of any number of nucleotides not divisible by 3. Misreading of all nucleotides downstream. Protein may be shorter or longer and its function may be disrupted or altered. We have discussed it. May occur due to slippage of DNA polymerase during replication. See, if this is the DNA polymerase, that is you need to replicate this strand of DNA. Now, what will happen is while transcription, see, don't confuse. This is a thymine. If there is a DNA replication, see, what will happen is during replication, you have DNA polymerase 3, which is the main, which is going on the leading strand. If you don't understand, kindly revisit the video on replication, so you'll better understand. Once during replication, if this DNA polymerase does not synthesize an nucleotide over a place, what will happen is there will be shift in entire chain, which is synthesizes the downstream. See, this is what is happening over here. The entire chain read on the downstream has been changed. It might alter the proteins that has been formed, or there will be no formation of proteins if they encounter a stop codon. These are seen in Duchenne muscular dystrophy, Tay-Sachs disease, cystic fibrosis. Okay. Splice site mutation, retain introns in mRNA, protein with impaired or altered function. Examples: rare causes of cancers, dementia, epilepsy, some types of beta thalassemia, Gaucher's disease, Marfan syndrome. See, we have in mRNA that in a primary transcript of an RNA, which is known as hrRNA, you have introns, you have exons, you have introns. Introns are nothing but intervening sequences to interrupt the protein synthesis. So these are removed by splicing. Let's say you haven't removed these by splicing because of splice site mutation. What will happen? These introns might get intervene with the protein synthesis and the protein might not be synthesized. This leads to diseases like cancers, dementia, epilepsy, beta thalassemia, Gaucher's, Marfan's. Okay. See this. Now we will go into examples. This is the coding sequence of DNA, GAG. Now, in silent mutations, what we have discussed according to Webber's hypothesis, the third nucleotide is we are not much bothered about a third nucleotide. See, either it's A, either it's not A, F, G, nothing. We are not much bothered. In missense mutations, what we are going see in silent mutation, there is only glutamine that has been synthesized. What might be the third amino acid, we don't care. In missense mutation, there is change in the second amino acid, second nucleotide. If the second nucleotide changes to thymine, this gives rise to uracil. When there is uracil in mRNA, it gives to valine. So the actual coding sequence which needs to do glutamine is changed into valine. So this is missense mutation. Okay. Now see. Now in coding sequence we have TAG, in which the guanine is mutated to thymine, which gives to uracil. When the uracil is there, you are gone. So it stops stop codon. When it is a stop codon, we have discussed it as nonsense mutation. Okay. In frame shift mutation, there are two types. Either there could be insertion, there could be deletion. What would happen? See, if you have thymine which has been inserting over here, if there is thymine, these three become one codon. Okay. So the new mRNA is formed, new aspartate. See, the glutamine is changed to aspartate. In frame shift deletion, there is deletion of a nucleotide which leads to an codon and which aspartate is synthesized instead of glutamine. Okay. Yeah. Next, going up to leucoporon. Leucoporon is a classic example of genetic response to environmental changes. Glucose is the preferred metabolite substrate in E. coli. But when the glucose is absent and lactose is available, the leucoporon is activated to switch to lactose metabolism. What I'm trying to tell is, generally, the basic metabolite which has been used by bacteria is glucose. Let's say you don't have glucose. What do and you have lactose. Now, bacteria need to survive on the lactose. To survive on lactose, they need the enzymes which can catabolize which can metabolize the lactose. Until when the glucose is present, this gene is not activated. because you have glucose. So there is no need of activating the enzymes of lactose metabolism and synthesizing them. So they will be suppressed. Okay, when you don't have glucose, when you have lactose, these genes will be activated, which gives rise to enzymes. See, now you'll see in low glucose increases. See, this is the mechanism by which all your lactose genes will be activated. When there is low glucose, there is increased and adenylate cyclase activity, increased generation of CAMP from ATP, activation of catabolic activator protein, which leads to transcription. When there is high lactose, there is unbinding of receptor protein from receptor operator site, increased transcription. Let's see the gene. This is a DNA gene which codes for your lactose metabolism enzymes. This is known as lactoprone. How is it uh, used when there is when there is low glucose and high lactose, or how to suppress it, or how do we enhance it according to the needs of the bacteria? See, once there is a decreased amount of glucose, let's see first there is a high glucose. When there is high glucose, the bacteria needs to utilize glucose, no use of lactose. Since it needs to utilize, then there is more amount of glucose, it inhibits adenyl cyclase. Adenyl cyclase is nothing but which converts ATP into CAMP. This CAMP binds to CAP. What is the CAP? Catabolic activator protein. When it attaches to the catabolic activator protein, it binds to the CAP site on the DNA. So when it attaches to the DNA, the promoter get activated. Once the promoter get activated, you have operator, you have lag Z, Y, A genes, which synthesize the respective enzymes for lactose metabolism. Not only you uh, see, yeah, okay. When there is high glucose, it inhibits adenyl cyclase, so this CAMP will not be formed. All this transcription will not be there. Once there is low glucose, adenyl cyclase inhibition is not there. So this adenyl cyclase can easily come and transform ATP into AMP, which leads to activation of CAP sites, which induces CAP sites, and that's how the transcription occurs. Not only you need the low glucose, but you need lactose. What's the use of lactose? This lactose will act as yellow inducer. What does this mean? Your lactose cell gene over here, it synthesizes repressor protein. This repressor protein binds to operator. Okay, when either either even if you have a low amount of glucose, it is making CAMP and it is activating CAP certain promoter site. This is not sufficient. You need extra. You need to remove this repressor to make to do transcription. So to remove this repressor protein from operator site, you need lactose. So what I'm going to tell is the glucose is required for transcription or lactose is required for removal of your repressor protein. So these are the different states in which your bacteria can be there. See, it have low glucose but lactose available. This is the state. This is the probability which we have discussed. When there is low glucose, there is increased CAMP, which activate CAP, which CAP activate the polymerase. Okay, here you have operator G. Operator combines with your repressor, but you don't need this repressor, so this lactose will bind to repressor. So you have enzymes coming from Z, lag, Z, Y, A genes. Okay, you have high glucose but lactose unavailable. See, you have high glucose, which means your bacteria will use glucose. What's the use? What is the sense of? There's no sense in synthesizing the lactose enzymes. So here there will be lactose unavailable, so there will be repressor protein binding to operator site, high glucose, so there is decreased CAMP, so there is no transcription. Let's see, there is low glucose and lactose unavailable. Since there is low amount of glucose, you have increased CAMP. If there is increased CAMP, capsid will be activated and no transcription. Because you need also lactose. If there is only lactose, then the repressor protein gets suppressed. Since there is lactose unavailable, this repressor binding is over here. So there is no downstream activity of a gene. Okay. Let's say you have high glucose and lactose available. Even if there is high, see this high glucose is inhibiting CAMP. So there is no activation of CAP site. So there is no activation of promoter. Although your lactose can go and bind to repressor protein, there will be some amount of activation of your genes, which leads to very low basal expression, either or no expression. Okay. So this is about lacoperon. Hope I cleared everything about lacoperon. Now going to eukaryotic gene. Functional organization of a eukaryotic gene. First, we will get the outline of what is DNA, how it is transcribed. See, once you have DNA, you have coding strand and non-coding strand. From the coding strand, you get your DNA gets transcribed into pmRNA. Is also known as hnRNA. Okay, so once this gets into pmRNA, it heads into splicing, mature mRNA, translation, protein. Before going into detail, I want to explain why there is need of synthesis of hnRNA. Why? Because you have, this is the nucleus. Say you have synthesized your mRNA over here. Here you have nucleolus. This nucleolus can degrade your mRNA. So it should not happen. So what we are doing is we are synthesizing hnRNA. See, this is your DNA. This is your first DNA, which contains coding strand. It has cat box, starter box, which is the promoter sequence, exons, introns. Then you have hnRNA, which is pre-mRNA. Then you have mature mRNA, which is forming the protein. Okay, so we will discuss in detail. Just give me some time. Regulation of gene expression, promoter, enhancer, silencer, epigenetics. Site where RNA polymerase II and multiple other transcription factors binds to DNA upstream, form from gene locus AT-rich upstream sequence with Tata and CAT boxes which differ between eukaryotes and prokaryotes. Promoters increase initiation of transcription. Promoter mutation commonly results in dramatic decrease in levels of gene transcription. See, we have 3 types of RNA polymerase. We will discuss in next page. They have given it. RNA polymerase 1, 2, 3. They are named in the order by which they have been synthesized. So first you have rRNA, which is nothing but ribosomal rRNA polymerase. It synthesizes the ribosomal rRNA, which is next is known as mRNA. This is tRNA. tRNA help in translation, not in transcription. Okay. See, site where RNA polymerase II and multiple other transcription factors binds to DNA upstream from gene locus, which is the promoter sequence, and this is the Tata box. Tata box. We have Tata cars in India. Enhancer DNA locus where regulator proteins activators bind, increasing expression of gene on same chromosomes. Enhancers and silencers may be located close to, far from, or even within a gene whose expression they regulate. Yeah. Silencer, DNA locus. See, enhancer and silencers are opposite to each other. This increases the expression of a gene, whereas this decreases the expression of a gene. That is the difference between enhancer and the silencer. Next, coming to epigenetics, changes made in DNA expression, heritable mitotically and meiotically. If there would a change in underlying DNA sequence. Primary mechanism of epigenetics change include DNA methylation, histone modification, non-coding genes. Let's revise once this epigenetics. What do DNA methylation does? Methylation mutes DNA. Acetylation activates DNA. What do you mean by muting a DNA and activating a DNA? Muting a DNA means there is decreased transcription of DNA or translation. Yeah, decreased transcription or replication of DNA. Here there is increased transcription or replication of DNA. Okay, that is epigenetics. The important thing that you need to note over here is they are heritable. Okay, we can inherit them from parents. So yes, this is important. RNA processing in eukaryotes. Initial transcription is called heterogeneous nuclear RNA, which is hnRNA. We have discussed. HNR is then modified and becomes mRNA. I have already discussed why we need to synthesize hnRNA first before synthesizing mRNA, because to prevent the degradation. See, nucleus, you have hnRNA over here, but the translation occurs in cytoplasm. So before to transfer this, to make translation, we need mRNA. So your hnRNA undergoes some changes to become mRNA. These changes is known as RNA processing. The following process occurs in the nucleus: capping of 5' end of 5' and addition of 7-methyl guanosine cap is co-transcriptional event. Polyadenylation of 3' and 200 adenosines, also known as polya tail, is post-transcriptional. See, this is co-capping. Polyadenylation is post-transcriptional. Splicing of introns, which is post-transcriptional. Capped tailed splice transcript is called mRNA. mRNA is transported out of nucleus to be translated into cytosol. See, I have discussed mRNA quality control occurs at cytoplasmic processing bodies, P bodies, which contain exonucleases, decapping engines, microRNAs. mRNAs may be degraded or stored in P bodies for further translation. See, I'll discuss. I'll try small boy over here. Sorry. Ooze. This is your mRNA. You have capping. This capping is called 7-methyl guanosine cap. This is co-transcriptional event. You have A tail. This is poly A tail. And you have removal of all your introns. Why we need to remove the introns? Because introns are intervening sequences. So your hnRNA becomes this is mRNA. mRNA by all these three changes: capping, tailing, and removal of your introns or exons, intervening sequences. Poly A polymerase does not require templates. Obviously, why you need a template? All you need is adenosine. There is no meaning in having a template. AUA polyadenylation signal, mutation in polyadenylation signal, degradation prior to translation. See, they have shown over here. This is the polyadenylation signal, which gives the signal to have a polyatyl. Okay, so basically this is your hnRNA. This is your mRNA. Next, Kozak sequence. Initiation site is in the most eukaryotic. This is important. It is either it is prokaryotes or eukaryotes. It is eukaryotes. Cossacks is present in eukaryotic mRNA, facilitates binding of small subunit of ribosome to mRNA. Mutation in sequence, impaired initiation of translation, decrease protein synthesis. Okay, Cossack sequence, what they do, they help in attachment of your mRNA to ribosomes so that they get translated. We will discuss now RNA polymerases. So RNA polymerase eukaryotes, we have already discussed. There are three polymerases: RNA polymerase 1, RNA polymerase 2, RNA polymerase 3. And they are numbered in the same order that their product are used in protein synthesis. See, first rRNA product is used because rRNA need ribosomes. This contains rRNA, and here you have mRNA. And after the third step is you have tRNA, which is clover shaped. We will discuss it. We have tRNA, which comes over here and attaches and undergoes translation. So this is the order by which we name all these RNA polymerase. RNA polymerase 1, rRNA. RNA polymerase 2, mRNA. RNA polymerase 3, tRNA. Alpha amantine found in Amantia phalloidosis death cap mushrooms inhibit RNA polymerase 2. What do RNA polymerase 2 synthesize? mRNA. causes dysentery, or have severe hepatotoxicity if ingested. Dactynomycin is an antibiotic, inhibits RNA. Sorry. Dactynomycin inhibits RNA polymerase in both prokaryotes and eukaryotes. Okay, dactynomycin is anti-cancer. Sorry, antibiotic, anti-cancer drug. Next, prokaryotes. Prokaryotes have only one RNA polymerase which makes all three kinds of DNA. Rifampicin, rifabutin inhibits DNA-dependent RNA polymerase in prokaryotes. They are used in TB. Okay. Coming to introns versus exons. Yeah, introns. Exons contain actual genetic information coding for protein and functional RNA. So we have retained exons in our mRNA. Introns do not code for proteins but are important in regulation of gene expression. So these are present in hnRNA. Remember, introns or intervening sequences. See, introns or intervening sequences stay in the nucleus, whereas exons exits are expressed. What do you mean by introns are intervening and inside the nucleus? Simple. They are present in hnRNA. They are intervening, which means they do not code for protein, and also these are present inside the nucleus. They are not transferred into cytoplasm, whereas your exons are transported across the into cytoplasm. Different exons are frequently combined by alternative splicing to produce large number of unique proteins. Okay. Alternative splicing can produce a variety of protein products from single hnRNA, heterogeneous nuclear RNA sequence. Example, transmembrane versus secreted Ig, tropomyosin variants in the muscle, dopamine receptors in the brain, host defense evasion by tumor cells. I'll add another example. You have B48, B100 in case of lipid metabolism. These are present in chylomicrons. These are present in LDL, VLDL, all these. So basically there is the same gene that has been synthesized in your B48 and the same gene that is synthesizing your B100. But the difference is, once you have intervened sequence over here, you have B48. Once you haven't intervened sequence over here, you have B100. That's the basic difference. See, DNA, hnRNA undergoes splicing and also capping and addition of poly A tail and undergoes mRNA, which undergoes translation and gives proteins. Okay, so we will discuss splicing in the next. Okay, we will discuss splicing now only. We will just discuss tRNA later on. Part of processes by which precursor mRNA, pre-mRNA is transformed into mature mRNA. We have discussed pre-mRNA is nothing but hnRNA transferred into mRNA. What is the meaning of splicing? We are removing the introns. Introns typically begin with GU sequence and end with AG sequence. Alterations in SNRNP assembly can cause clinical disease. Example, spinal muscular atrophy. SNRP is important. Spinal muscular dystrophy is produced because of the mutations in SNRNP assembly. SNRNP assembly is affected due to decreased spinal muscular nuclear protein, which leads to congenital degeneration of anterior horns of spine cells, symmetrical weakness, hypotonia, floppy baby syndrome. SNRPs are snRNAs bound to proteins, Smith proteins, to form spliceosome that cleaves the pre-mRNAs. These are nothing but your snRNAs which are bound to proteins which are Smith proteins. Anti-U1 snRNP antibodies are associated with SLE, mixed control disorders, and other traumatic disorders. We have different antibodies that are formed against these spliceosomes. Once these antibodies attack the spliceosomes, we get something like SLE, we have mixed control disorders, we have dermatomyositis, everything will be formed because of this. See, this is exon, this is intron, this is exon. We need to remove this intron. How do we remove this intron? You have U1 SNR1 snRNP. This is U2 snRNP. These are snRNPs are nothing but RNAs with small proteins which might be Smith proteins. Once they attach over here, see, they are clumping all these introns. They are removing this and making it as mature mRNA, which is exon, only exons. This is introns. So that's how your transcription occurs. We have discussed this. Okay.